Welcome, welcome, everyone. Thanks for joining us at NU at Noon. This is actually our second session, thanks to those of us who joined us last week at the tour of EXP. It's a beautiful building. If you were not able to join us, I highly recommend sneaking onto campus and checking it out. My name is Gina Reganini. I'm the Director of Educational Initiatives in Alumni Relations, and I'm so excited to share this day with you. Before we dive into it, I wanted to get a couple of housekeeping items out of the way. First is I want to extend a huge thank you to the NU at Noon committee. They are a fierce bunch, and none of this programming would happen without their support and their insight and their suggestions. Those folks are Ann Wilson, who's here today, Brian Doherty, also here today, Angela Marcucci, Joe Hunt, Marguerite Krupp, and Ken Zuchano. So thanks again, everyone, for your support. If you're interested in being a part of the committee, please do email me. My other name is the one who's been emailing you, so you no doubt received a message from me at some point over the last couple of weeks, so please feel free to be in touch. The other piece that I wanted to share are a couple of upcoming exciting events that we've got going on at Northeastern and in Alumni Relations. The first is this coming weekend. As you may or may not know, it is Head of the Charles weekend, and Northeastern will have a tent. So if you are planning to be at Head of the Charles, please come find us. We'll have some chocolate bars and some swag and some goodies, so please come by and say hello. And bring your umbrella because Saturday looks a little dicey. And then in November, from November 10th and 11th, is homecoming weekend. So we'll have a bunch of exciting events going on that weekend, including the return of beloved Punter's Pub, this time in a trailer, but it looks exactly like the real thing, I promise you. So I hope you'll join us for that as well. But now let's get into our presentation. I am so thrilled to welcome Dr. Nikolai Slavov. He is the Associate Professor of Bioengineering and also the Founding Director of Parallel Squared Technology Institute. He received his undergrad at MIT and a doctoral at Princeton. He's been at Northeastern for 8 years, and we're so excited to have him here with us today. Thank you, Dr. Slavov.
Thank you for the kind introduction, and thank you very much for joining us today. I'll share with you ongoing research, both from my laboratory and from the laboratories of colleagues, on understanding the pathology of Alzheimer's disease. It's a terrible disease whose pathology, whose causal mechanisms remain very poorly understood, and this lack of understanding fundamentally limits our ability to develop effective treatments. As perhaps all of you know, it's a very common disease. It affects more than 6 million Americans at the moment. And as our population average population age increases, it's projected to affect more than 13 million individuals by 2050. It's also a very expensive disease in terms of care for loved ones, in terms of families, the way it impacts the families, and its ultimate effect on our healthcare system. Almost everybody, every gender, ethnic group, every country in the world is affected by Alzheimer's disease. Some cases, a small number of cases, are familial cases, and those familial cases are primarily caused by mutations in individual genes, but that accounts for only 5% of all cases of Alzheimer's. The vast majority of cases are called sporadic cases, and that means that there isn't a single gene that causes it. There are a variety of factors, both environmental, genetic, lifestyle, and so on. And while it affects both sexes, it is more common among women, almost twice as common among women compared to men. From the very beginning, from the first discovering and naming of the disease in 1906, there were two very obvious pathologies associated with the disease, and those are protein aggregates, either extracellular amyloid beta, commonly known as A beta plaque. These are outside of the cell, intracellular tau neurofibrillary tangles. At the time when Dr. Alzheimer first made those observations using a simple light microscope, the molecular nature of these plaques wasn't known, but by now we know very well that their major components are these proteins, A beta and tau. And what we have observed over the decades is that A beta plaques tend to occur earlier, and their presence precedes the development of clinical symptoms. It precedes the development of memory problems, dementia, and the other clinical symptoms associated with the disease. And they also develop in areas of the brain that are less tightly associated with the cognitive dysfunction. While those neurofibrillary tau tangles are much more closely associated, much more closely correlated with the symptoms of the disease. And that's why we're going to focus more on the tau protein. It's a protein with not well understood function, and it's a protein that might exist in a variety of different conformations, so it can be soluble as monomers or oligomers. Oligomers mean multiple protein molecules or aggregates of more protein molecules. And here I present pictorially some predicted models of what can be the structure of soluble tau protein, but there are many more structures that exist, some of which with unclear significance and not well elucidated 3D structure. And it's clear from this very brief introduction into Alzheimer's disease, while we don't know what's really causing it, from the beginning it has been clear that proteins are at the crux of the disease. They're tightly associated with it. And therefore, to study Alzheimer's disease, to understand it, it's useful to be able to analyze proteins, their modifications, their age, their turnover, how they interact with other proteins. And here I've made a brief diagram of what are that we have to analyze proteins. One set of methods based on affinity reactions or antibodies, so just recognize a particular protein of interest, and then that antibody is conjugated to some molecule that allow us to detect it. And those antibodies are similar to the antibodies that fight diseases. They're part of the immune system, but in this case, they're specifically developed to recognize an epitope. Epitope means a small piece of the protein, to bind it specifically, and then report on the presence of that epitope. The challenge with these antibodies is that they don't only bind their specific spot, they also bind lots of other proteins, and this results in low specificity. And they don't detect the whole protein, they don't detect all of its modifications, they just detect that one spot. And furthermore, it is very challenging to analyze many proteins simultaneously with these antibodies. People have not been able to analyze more than a few dozen, a few hundred at most, while there are very many proteins. There are tens of thousands of distinct proteoforms. And another set of methods that my research has pioneered and is focused on is using liquid chromatography mass spectrometry. And I'll give you a brief introduction to what that is. But before I dive into that, I just want to share one of the problems from global perspective with antibodies is that their quality tends to be rather variable and oftentimes rather low. So Nature has published an estimate 8 years ago that annually almost $1 billion on antibodies that do not have the specificity indicated on the label. So the reagents that are produced and sold to detect the protein don't detect that protein. They detect something else, and we don't know about it. Major problem. So that's why we really need new and better tools to study proteins that are ultimately at the crux of the disease. And the most powerful tool that humans have developed to study proteins globally, specifically with precision, is mass spectrometry. And mass spectrometry in its fundamental essence, in its concept, is basically measuring the weights of molecules very precisely and very accurately. And then we break the molecule into pieces, and we may measure the weights of those pieces, and from the weights we can put together what the original molecules were. Here you can see a diagram of different protein molecules that then are usually digested into smaller pieces, peptides. And then we can use a process to separate them in time. It's called liquid chromatography. It's just a way to sort them out so that similar proteins come out at about the same time. Then we can measure their weights. We measure the weights with accuracy of error 1 per million. Very, very accurately can measure their masses of small molecules, and then we break them into pieces, and from those pieces we can put together the puzzle from their masses. So that's in essence. This is a slightly more technical explanation. This is real data, where here on one axis we have the separation as those peptides are coming out from the liquid chromatography. On the other axis, we have accurate measurements of mass over charge. M over Z is mass over charge. And here we can see individual peptides, which are groups of peptides, which are then broken. These are the pieces of one peptide. And from these data, we can determine exactly what the protein is. OK, so when we do that, and we analyze tau proteins from human brains, we can detect many, many dozens of modifications on the tau protein. So different amino acids of the protein get modified. They get either phosphorylated, they can have acetylated. So these are small molecules that are attached to the protein. And these small molecules attached to the protein can change its conformation, can change its shape, and can change its dynamics and functions. And the question, of course, is it's great to have this technology to be able to analyze the modifications on the protein, but does it matter? Is it associated with the pathology? And here is a short snippet of data that correlates the presence of such modifications on tau with its seeding, meaning how able it is, how able those molecules are to generate the development of tangles inside of in this case, experimental mice, or to what extent they cause pathology here on the right with the disease progression. And we see that there is a substantial correlation, a strong correlation between the modifications present on tau molecules and the ability of those proteins to cause the disease, both to generate the tangles and also the rate at which the disease progresses. And this really provides a compelling motivation for us to delve more deeply and to study proteins, but not only study them across a dozen patients, but be able to study them across many hundreds and many thousands of patients so that we can identify robust associations. We can understand what's happening. And this motivation is what led to the establishment of a new research institute, Parallel Square Technology Institute, that we started earlier this year, that is developing much higher throughput protein technologies for doing these analysis of Alzheimer's disease. And I'm going to briefly convey to you what are some of the ideas, what are some of the concepts behind the ability to increase the throughput of the analysis. Of course, you have heard that much of biomedical research for a long time has been focused on analyzing genomes and DNA. In part, this is because this is much easier to do. We have existing technologies. NIH and other major funders have invested billions of dollars to make the analysis of RNA and DNA molecules very inexpensive. And as a result, we can associate genetic polymorphisms, changes in DNA to the disease. But the problem is that these associations are very indirect. As we analyze the DNA changes don't really tell us how the disease developed. They just tell us that there's some distant correlation. And because those technologies are so available, most researchers use them. They use what they have. And it's a little bit like looking for the keys under the lamp post, not because that's where they were lost, but because that's where the light is, which in some some ways is sensible thing to do. But there is an alternative approach, which is let's build a new, much more powerful lamppost so that we can shine light where the keys were actually lost. And that's what we're trying to do in the case of proteins. I here show this diagram from a recent review from colleagues. For the scale of increasing complexities, we go from genes in the genome to RNAs that are transcribed to the genome, to proteins and interactions between proteins. We have tremendous explosion of complexity. One gene can result in hundreds, even thousands of different proteins that have lots of interactions with different proteins. So understanding biology at the level of the proteome is substantially harder, and our technologies for that have been traditionally much more limited. The most powerful one being mass spectrometry, which I already introduced, but mass spectrometry has had relatively low throughput and has been relatively expensive, which means that it's difficult to analyze sufficient samples to power studies. So our goal is to make mass spectrometry analysis of proteins thousands of times faster and cheaper. And then we want to use it to identify these protein aggregates that are at the root of the disease. We want to be able to analyze monomeric, oligomeric, aggregated forms of protein. We want to be able to see their modifications, how they change their conformations and their interactions. And ultimately, we want to be able to do this across sufficient number of patients so that we have well-powered discoveries. We don't just have a hypothesis that next week somebody else measures a few more samples and they find that it doesn't hold. We want to have a robust data set at scale that can robustly constrain the causes for the disease. And conceptually, the way we approach this problem is by parallelizing the analysis of samples and parallelizing the analysis of peptides. So I told you that with mass spectrometry, the way we analyze proteins is by weighing the protein, then we break it down and we weigh the pieces. And we can do this for one sample and one protein or one peptide at a time. Or we can figure out a way how we can label samples from many different people or different parts of the brain, and then once they're labeled and they're made distinct so that you know each one came from that individual, we can mix them together. And on the other axis, instead of analyzing one peptide or protein at a time, we can start analyzing hundreds, and this is another axis. So if we combine both of these, we get a multiplicative scaling so that per unit time, we can start analyzing many, many more proteins. And what is important here is that the cost decreases proportional to the throughput, because the cost is really the expense of the instruments. The mass spec instruments that are doing these experiments that can weigh a protein molecule with exceeding precision and accuracy are naturally quite sophisticated and pricey. So if we're able to analyze thousands of samples more per unit time, then it becomes thousands of times cheaper to do that. And this is something that we developed in my lab and validated. It's reported in the scientific publications. I'll just show you some data to give you a sense of how the raw data from this analysis looks like. So here are measurements from single cells using this method. And we analyzed 2 different human cells, pancreatic adenocarcinoma or monocyte cells. And here we can see the measurements of the peptide ions. This is a specific sequence from this protein. And then here are the fragments. As we break the peptide into fragments, we can also measure the fragments. And then we can quantify the abundance of this protein between these 2 different cell types. And we see that our estimates of the abundance of this protein are highly consistent from the different data points at the level of the peptide ions, at the level of their fragments. And this consistency allows us to have confidence in the data quality. And of course, we can zoom out from the example of this one protein, and we can ask how does it compare if we do this analysis on a more global scale, comparing measurements of protein abundance by this method or another easier, well-established approach of larger samples. And we see that across hundreds of different proteins, we obtain very similar estimates for their abundance, which gives us confidence that this method works, because as we try to increase the rate of data generation, as we try to increase the throughput, we want to preserve the quality, the quantitative accuracy. We don't want to sacrifice those aspects. I think this is actually a good point to maybe pause and ask for questions if what I'm saying makes sense, or if you want me to elaborate more on something that I said. Instead of me keep talking, I'm happy to do that. I can talk for hours. I have a lot of experience with it, but I think the beauty of me being here with you is that we can also have a conversation. If there is something that you're particularly interested that I said, I'm happy to address that more. We're going to have more time for questions at the end. I just wanted to make sure that also in the middle I break down and give you the opportunity to ask questions.
This microphone is for all of our friends who are joining us online. So if you do have a question, I'll come over and bring the mic to you.
Yeah, my question is just, the problem seems to be within the proteins is the amount of folding, but don't proteins do that anyway?
So every protein folds, at least to some degree. Some proteins fold in more rigid shapes than others, but any one specific protein, such as tau is a good example, can fold in thousands of different shapes. And then tau is not one protein because it has all these modifications. So if we go back, here on this axis we have the amino acids, the building blocks of the tau protein. And at each of those positions, the protein can have those different modifications with the abundances shown here on the y-axis. So tau, we think of it as being one protein, but it's one protein that has hundreds of different guises. And each of those modifications can make it fold in a different way, or even the presence of other proteins can make it fold in a different way. So even if it's the same protein, it can have very different function. And probably the best known canonical hallmark example of this are prions and mad cow disease, where we have a natural protein that is normally present in our brains that can change its conformation, fold in a different way, and causes a disease. And that's why it was so incredibly difficult for researchers to work out exactly what's happening, because they were confused. How come the disease is caused by our own proteins? At the time, the majority of people believed that mad cow disease and other diseases were caused by viruses. And they were looking for RNA and DNA, and they couldn't find RNA and DNA. And the people who were prominent researchers in that field, such as Stan Prusner, was leading the research, and he kept saying, I don't see a virus. And he came up with a different name, and the whole scientific community couldn't believe for decades. They were going back and asking, there must be a nucleic acid. No, there's no nucleic acid. So it came to the point that when he received his Nobel Prize, the main newspaper in Sweden published an article, has the Nobel Committee made a mistake? And at that point, the scientific establishment knew it wasn't a mistake, but just giving you a nuance of how counterintuitive and confusing it was. By now, it's very well established that prions are really the same protein folding into different shapes. And what we have in Alzheimer's disease is similar, that we have a protein that can have folding into the right shape and can also fold in many different shapes that can be pathogenic and contribute to causing the disease. And one complexity with this is that even when we look at associations like these ones, where we know that what are the modifications of the protein and how these modifications are associated with disease progression, and we see strong correlation, that seems really useful observation. It is, but it's difficult to say it's causal. It is difficult to say from these data that simply because we have this correlation, those modifications are what cause the disease. In this case, the way this experiment was done, because it's an interventional experiment, we are a little bit closer to being able to make this conclusion, but it's not a definitive proof, let's put it that way. But wonderful question. Thank you for asking. And the more we can make this as conversation, as me answering your questions rather than me telling you what I do, the better. That's why I'm here. I would love to answer more questions on anything that I mentioned or things that I didn't mention. If you have questions, please send them my way. I know that what I'm explaining, especially all this mass spectrometry business, that's quite technical and complex. I try to make it simpler. It is fundamentally about weighing molecules and then breaking them into pieces and weighing those pieces, and then we have to put them together, which is a little bit like putting together a puzzle, where the different pieces of the protein correspond, or peptide correspond to the pieces of the puzzle. And then what we are doing with this parallel analysis that I'm telling you is that instead of breaking one puzzle at a time, we can break hundreds of them. And then, of course, putting them together is much harder. But if we can do that well, then we put together hundreds in the same amount of time that previously we did one. And the technology, the mathematical approaches and algorithms that we helped develop made that possible. And this is why now we can start increasing, using this framework, we can start increasing the number of samples that we can accurately analyze, quantify per unit time, and decreasing cost. And this is what makes it possible for us to start analyzing at much larger scale the pathology in humans. And there is also, I told you a bit about how we measure proteins, but of course there is a question, how are you going to introduce those proteins into the instrument to analyze? How are you going to prepare the samples in technical jargon? And we prepare the samples by isolating individual cells in these droplets. And each droplet can receive various reagents and be prepared in parallel. So here I've recorded for you a movie of one of the steps of this sample preparation process. And hopefully this plays. It opened it in the browser. Rather than, that's okay. I think it will be harder. This worked well in the previous, in Google Slides, but now that this is being ported into PowerPoint, it's harder to play it. I just wanted to show you a video of how this sample preparation works. But essentially, it's a fully automated robot that is able to individually address each of these droplets, and each of these droplets contains a sample from a single neuron or a few neurons from a brain where we know the position, and we are able to prepare many thousands of those in this automated way and then apply the highly parallel analysis. And developing this initiative as part of a nonprofit research institute, Parallel Squared, because we think that this is an approach to maximize the impact. Instead of generating data that only one company can hold up and use for its own developments, we want to share the data with the broader community so that we can empower more people to test their hypotheses and to figure out what might be causing Alzheimer's disease. And we want to be able to develop the technology itself for this parallel analysis that I mentioned. It's something that would very much benefit others being able to use rather than being developed just in proprietary settings. And again, we have the objective to share all of that. And this was our motivation to develop it as a nonprofit research institute rather than doing it for profit. We have been very proactive in sharing our protocols, code, data, and building data portals. So this is one example for a data portal built at Northeastern for a lab from my lab. And also developing guidelines for the rest of the community, people who want to adopt our methods, sharing best practices of how to do that. And finally, I want to tell you about something that is really hot off the press. It is very cutting edge, and that very, very few people in the world know about it. It's a very surprising observation that we made. So perhaps some of you know the idea of the central dogma, the genome encoding what sequence our proteins are going to have. And this has been our understanding for the last 50 years, ever since the structure of DNA was elucidated. And this, by and large, has held, and we have a lot of evidence that this is true in many cases. But what we were able to do is to ask how frequently the amino acid sequence of a protein in our bodies from what is encoded by the DNA. Here I'm showing you one example of a short sequence, so each letter corresponds to an amino acid. And in this case, we can have in this particular place of the protein, instead of having T, we have G, a different amino acid being inserted. And we figured out that there is a way using this very precise weighing of peptides and their fragments to efficiently discover those substitutions, cases where we have a different amino acid. And then we can apply various downstream validation approaches to make sure that we can trust this observation, this data. Now, why could an amino acid be different? Would you expect ever to observe that? The answer is yes. Nothing in the world is ever perfect. When we make things in our manufacturing facilities, we make mistakes. When our bodies make us, they make mistakes. All the time, those things do happen. But they generally happen at relatively low level, maybe 1 per 100, maybe 1 per 10,000, 1 per 100,000. And there are various mechanisms to cope with those inevitable mistakes that would happen. And we wanted to see, that was really the intention, for the first time to globally be able to quantify this at the level of the proteome of making proteins. And then, so here is a diagram where we have the messenger RNA being translated by a ribosome and making proteins. And here is the protein that is templated by the RNA. And here is a protein that has a single amino acid being swapped. And we can ask how efficiently these proteins are, what is going to be the relative abundance. And the relative abundance is going to depend both on how quickly we make this protein and also on how quickly we degrade it. Because each protein molecule has a certain rate of synthesis and then has a rate of degradation. And ultimately, how much we have depends on how quickly it's made, how quickly it's degraded. It's a little bit like a water tub. How much water you have in the water tub depends on how much comes in and how much leaks out, and those two processes. So what was the really bizarre, surprising observation that we made in doing this analysis is that when we looked at the abundance of those modified proteins, most of them, with amino acid substitutions, most of them were very lowly abundant, and that's what we would expect. That's not so surprising. But some of them, about 10% of them, were more abundant than the protein templated by the RNA. And we thought that's really unexpected and strange. And we spent 2 years trying to validate it with various approaches. And I'm going to spare you all these technical details. It becomes quite involved. But ultimately, the evidence was such that we couldn't ignore it, and we thought, well, how could this possibly be the case? And the model that we came up, what we think is happening, is that while this protein that has sequence templated by the RNA is produced much more quickly, much more efficiently, just as the central dogma would tell us, every now and then, the mistakes that are being made can destroy the region of the protein that targets it for degradation. So recognized and say this protein should be degraded. Now, if you substitute the amino acid that participates in this targeting, you can make the protein much longer lived. And this is what we think happens with some proteins in this case, that we have the substitution. Even though this protein is maybe produced at 100 times lower rate, now it becomes very long-lived. It's not being recycled. And this leads to that protein with an amino acid substitution, which can potentially alter its function. It's going to hang around for a long time. And we think that this kind of process, which is really a new discovery, we didn't know about it, we think that this would have big implications for all sorts of diseases, especially aging-related diseases, because those things are defective parts made by my body that hang around longer than they're needed. And this, we think, is a very exciting new avenue to investigate, and also investigate what are the implications of this kind of stabilization of an amino acid substituted protein in the context also of Alzheimer's disease. So the big surprise here is the observation, not so much that we have those proteins with amino acid substitutions, but that they're so highly abundant, and so many of them. I'm going to finish by thanking the wonderful group of PhD students and postdocs and undergraduates in my lab at Northeastern University that have made all the work that I had the privilege of sharing possible. And not only possible, but has made it a great learning experience, a lot of fun for both me and them. And I would like to thank our sponsors, which are both the NIH Directors Award, which is federal funding, and also a number of philanthropic organizations. The Paul Allen Frontiers Group gave me an Allen Distinguished Investigator Award, which is a high-risk, high-reward type of funding that enables to break new grounds and do things that are very different from and very challenging from what has been done before. And Schmidt Futures, the philanthropy of Eric Schmidt, supported the establishment of the new institute, Parallel Squared, that I mentioned, for scaling those technologies. I'm going to stop here, and I'll be very happy to answer more questions if you have questions for me.
Any other questions in the room? And it looks like no questions online. If you're joining us on Facebook, feel free to throw a question in the comments section. This is fascinating. Yes.
Could you give a sense of what the primary outcomes you would expect to see from your research or from the field in, say, 5 years from now and, let's say, 10 years from now? What is it that you're aiming to accomplish that gets closer to either a cure or at least some treatment for Alzheimer's?
So there are different avenues of focusing on cures for Alzheimer's disease. Some of them are very short-term ones that say we don't really know what's causing the disease, but we're going to start doing something and hope that we're going to find out an effective treatment. An example of that kind of approach is the development of antibodies for some of these protein aggregates, such as A-beta. And there have been clinical trials funded with antibodies that successfully target A-beta, and people eventually found that this is not effective. It doesn't help patients. And there are ongoing efforts of this kind where people are developing antibodies without being certain in what is the mechanism of the disease. And these are the really high-risk investments on the side of biopharma that, if successful, are going to be the ones that give the fastest results, potentially in time frame of 5 to 10 years, to your question. But many of those can have side effects because we don't exactly understand what we are targeting, and they can also have the fate of the antibodies for A-beta because they're targeting something that may not be the right target. What I'm focusing within my research in the institute is really on understanding the causal mechanism in different patients, being able to better diagnose it, and then once we know what is the cause, being able to directly target it to minimize side effects and to have high confidence that this is going to be effective. So my own hope for what I would achieve within the next 5 years within my research teams is that we would have a well-supported model for what is causing Alzheimer's disease, so that we better understand the disease pathology. What are the mechanisms, for example, of neuroinflammation? We know that neuroinflammation contributes to the disease, but we can figure out what are the signaling mechanisms, what are the pathways leading to that neuroinflammation, so that we can mitigate it. And that's potentially something that can have very real, fast, tangible, positive impact on patients, as subsiding, reducing the neuroinflammation can decrease the rate of neuronal death and can quickly alleviate symptoms. That would be one possible therapeutic avenue. Another one is understanding how to prevent the pathological aspects of these protein aggregates, which are the exact modifications that are associated with the disease. So here is some of the data that they showed directly speak to that. We see that the tau protein has lots of different modifications. Each of these bars is a different modification. And if we're able to do experiments like this to identify which are the modifications that are best associated with the disease, and not just correlated but causally associated, then we can direct interventions towards these modifications so that they can be more effective. So the primary outcome within a 5-year time frame for us is going to be this understanding that provides specific direct targets for effective therapies with minimum side effects. Yes, I see a question.
Right now, is it only found by diagnosis of when you see the patient?
Yes. So usually patients who start having memory problems would be diagnosed as some form of dementia. There are many different types of dementia, not only Alzheimer's disease. Alzheimer's disease is by far the largest cause of dementia worldwide, certainly in the US. So if patients are having memory challenges, they're likely related to AD, but they may not be. And then they go and visit the physician, and there is a whole battery of clinical tests for the diagnosis. It depends on how progressive it is and the type of memory loss that is present, and there are also brain scans that can be performed to detect the accumulation of plaques of A beta and tau neurofibrillary tangles, and based on that information, the physician would diagnose or not diagnose AD.
So, why do you hope to be able to get it through a blood draw? Or
that's right. There's certainly a hope that we can do the diagnosis faster, simpler, easier, to do it earlier, at earlier stages. So there are some observations that these pathological forms of tau that are present in the brain can also be detected in cerebrospinal fluid. And cerebrospinal fluid can be sampled less invasively than sampling the brain, which of course we cannot do with a live human patient. Usually these measurements that are done on human tangles and A beta, they're post-mortem. But to the extent that these proteins are present in the cerebrospinal fluid, then these kind of mass spectrometry measurements that we do, protein analysis, can detect them. And then they can certainly help with earlier, more reliable, simpler diagnosis as well.
Any other questions? Wonderful. Dr. Slavov, thank you so much for joining us today. This was really wonderful. Congratulations on your $50 million grant and for the time you spent with us today.
Thank you.
Thank you.
I'm happy to be here.